Good day. I, along with my group mates, will be presenting the content of our experimental design, which deals with the spectrophotometric analysis of aqueous methylene blue solution. Most test substances in water are colorless and invisible to the naked eye. In this experiment, aqueous methylene blue solution is used. Methylene blue is a bright greenish blue organic dye which is primarily used in baths and to a lesser extent of paper. To test any presence of test substance which is itself colored or can be reacted to produce a colorimeter or a spectrophotometer can be used. So, what is spectrophotometry all about? Spectrophotometry is the quantitative analysis of the interaction of ultraviolet, visible, and infrared radiation with a substance as a function of wavelength. The nature of this interaction deals with the physical properties of a material, such as its opacity, thickness, roughness, smoothness, and even pureness. So, with the use of spectrophotometer, which is an analytical instrument or device used to quantitatively measure the visible light, ultraviolet light, and infrared transmission or reflection, then the method of spectrophotometry can be done. Some applications of spectrophotometer includes the detection of concentration, detection of impurities, solution elucidation of organic compounds, and monitoring the oxygen content in water and marine ecosystem.
So, how does a spectrophotometer work? As we can see in this image, first, a light strikes the diffraction grating, which acts like a prism and separates the light into its component wavelengths. The grating can be rotated so that only a specific wavelength of light reaches the exit slit. The light will then interact with the sample, and the detector measures the transmittance and absorbance of the sample. The detector senses the light being transmitted through the sample and converts this information into the digital display or meter. So, if we collect this data continuously over the duration of a chemical reaction, we can gather the changes in absorbance and the change in concentration of a substance over time. In order to do this, we will use the Beer-Lambert law, which relates the amount of material in a sample to the absorption of light passing through it. This law helps us to describe the quantity of light absorbed and transmitted. According to this law, the amount of light absorbed is directly proportional to the amount of solvent. Here, A is for the absorbance. The epsilon is called the molar absorptivity, which is a measure of how well a chemical species absorbs a given wavelength of light. L is the distance that light has to travel through the sample, and C is the concentration of the sample. Absorbance is measured by comparing the intensity of the beam of light as it enters the sample, and the intensity as it leaves the sample, as we can see in this image below, which are I0 and I respectively. Here, A is equal to negative log I over I0. So the objective of the experiment is to determine the absorption spectra of methylene blue. A line equation should also be accomplished because it describes the relationship between the concentration and absorption of the sample. This line equation will help in identifying the concentration of methylene blue in the solution.
We are now moving forward with the methodology for this experiment. The experiment is pivoted in the determination of the absorption spectra of a methylene blue sample, the line equation that would relate absorption and concentration, and the concentration of one sample of methylene blue. Ascertaining the absorbance spectrum of the samples will be done through the process of spectrophotometry. Meanwhile, the line equation will be derived from the samples with known concentrations and absorption values. Lastly, through the analysis of the trend line, the methylene blue sample with an unknown concentration can finally be deduced. To summarize the path that we need to take, we have to first find the absorption spectra of every sample. Secondly, through known concentrations and acquired absorption spectra, we can then create the line equation to correlate the aforementioned variables. And lastly, through our line equation, we can then determine the unknown concentration of the methylene blue sample. Pushing forward, the reagents necessary for this experiment includes prepared samples of methylene blue and water. On the other hand, for our instruments, we will be utilizing the Spectronic 20 spectrophotometer, a device that is employed in order to gain the measurement of absorbance or transmittance of solutions. Also, other lab equipment such as test tubes and pipettes are necessary in transferring and holding reagents. Let us now dive into the procedure of the experiment itself. To begin with, the Spectronic 20 spectrophotometer should be turned on and warmed up for around 15 minutes before the experiment proper. Meanwhile, to prepare the measurement of the absorbance spectrum, two test tubes must be prepared. One that contains 5 ml of the stock solution of methylene blue and another that contains 5 ml of water. When the machine is already warmed up, the wavelength must be set to 400 nanometers. Subsequently, the machine must be zeroed. With the lack of tube in the machine, the zero control must be turned on in order to change the needle into a 0% transmittance or infinite optical density, what we call DOD. Afterwards, the blank tube must be put into the sample chamber with the chamber's cover closed and the transmittance or absorbance knob adjusted just exactly when the needle lies in the 100% transmittance. Laying out the actuality that the machine is properly warmed and zeroed, data for the absorbance spectrum can then be accumulated. With a wavelength set to 400 nanometers, the tube of the stock solution must then be placed in the sample chamber. Its OD must be recorded. After recording the data, the tube must be removed and the wavelength must be adjusted to 425 nanometers while zeroing the machine all over again. Similar to the first step, the OD of the stock solution for this adjustment must be recorded. The same arrangement of steps, the adjustment of the wavelength by 25 nanometers, zeroing the machine, and recording the OD of the stock solution must be repeated 21 times until the readings for the same stock solution across a vast spectrum of wavelengths is gathered. On the other hand, to find out the standard curve of absorbance versus concentration, certain steps are needed to be taken. Firstly, serial dilutions of the stock solution should be prepared. Meanwhile, through the use of a pipette, a 5 ml of the stock solution must be placed in a test tube in the absence of water. Accordingly, a second test tube must then be filled with a 4.5 ml stock solution and a 0.5 ml of water. This arrangement must be proceeded for a total of 9 test tubes. In addition, it must be noted that the wavelength is held constant for this part of the experiment. The wavelength utilized is the peak wavelength from the previous segment, which is 625 nanometers. With this setup, the machine must be zeroed for this wavelength and every dilution tube must be situated individually in the sample chamber. The OD must be recorded. Together with this data, the molarity and the corresponding concentrations are obtained and calculated. The aforementioned acquired data must then be plotted in an XY scatter plot in order to find out the best fit line. Lastly, the tube with the stock solution of an unknown concentration must be placed in the sample chamber without any kind of adjustments. When the standard curve for this wavelength is established, the sample with an unknown concentration must be plotted on the best fit line through the use of the OD. With this, the correlating or corresponding concentration can be deduced. To tie everything into a notch, this followed process is modeled to the experimentation of Danelli et al. In 2006 entitled Introduction to the Spectrophotometer, Wavelength, Absorbance, and Concentration in Methylene Blue.
As established earlier, this experimental design is actually modeled after an already existing experiment, which is why for this presentation for the results and discussion, I will also be presenting the results that they have gathered. It is important to note that these results may vary depending on what you will obtain in your own experimentation. So for objective number 1, the determination of the absorption spectra of the methylene blue. As we could see in table number 1, the absorbance of methylene blue at various wavelengths. In the first and third columns, we could see the wavelengths of the light that is shot through the cuvette. On the other hand, the columns beside them are the absorbance reading from the spectrophotometer. Meanwhile, the figure on the right is actually a graph, a plot of the data that was tabulated in table number 1. Looking at the data and its corresponding graph, we could observe that the absorbance is increasing from 400 nanometers to 650 nanometers. In here, the absorbance is directly proportional with the wavelength, which means that as the wavelength increased, the absorbance increased as well. We could also observe that the absorbance peaked at 650 nanometers with 0.975 optical density. Lastly, as the absorbance has reached its peak at 650 nanometers wavelength, the absorbance started to decrease from 650 nanometers to 900 nanometers. In here, the absorbance is inversely proportional with the wavelength, which means that as the wavelength increased from 650 nanometers, the absorbance decreased. Comparing the experimental peak absorbance of 0.975 OD to the literature peak absorbance of 668 nanometers according to Huang, Xie, and Chen, there is a 2.69% error, which implies that the experimental value is fairly close to the literature or actual value. Looking at table number 2, we could see that going from sample 1 to 9, the concentration is actually going down. This is because of what was mentioned in the methodology, wherein the ratio between the stock and the water is changing for every sample, wherein in sample 1 there is a high concentration of the methylene blue, while at the last sample there is little concentration of methylene blue. Looking at the absorbance of the samples, we could see that it is actually decreasing as the concentration decreases. This is further illustrated in the figure next to it in the graph concentration versus absorption of the methylene blue. We could see here that as the concentration of the methylene blue increased, its absorption increased as well. of Microsoft Excel, the line equation was obtained to be y is equals to 20,476x plus 0.0211. The line also has an R-squared value of 0.987, which is an indicator that there is a strong correlation between concentration and the absorbance of the methylene blue solution. This directly proportional relationship between the concentration and the absorbance of the solution is actually in agreement with the Beer-Lambert's law that was discussed a while ago, which states that the absorbance of a solution is directly proportional to its concentration. So the equation y is equal to 20,476x plus 0.0211, we could manipulate it so that the x, the concentration, is alone at the left side. So we have x is equals to y minus 0.0211 all over 20,476. From the reference experiment, the obtained absorbance of the unknown sample was actually 0.220 OD. Plugging that into the equation, we obtain a concentration of 9.76 times 10 raised to negative 6 mole per liter. So what if you conducted the experiment yourself, and when you subjected the unknown sample to the spectrophotometer, you obtain an absorbance of 0.540 OD. Once again, you just plug that into the equation, and you will obtain a concentration of 2.53 times 10 raised to negative 5 mole per liter. So now that we have met the objectives of this experimental design, what could be its possible sources of error? So first, it is possible that the fingerprint of the handler or some foreign material stuck to the surface of the cuvette, which may not have been wiped clean completely. These obstructions and markings on the cuvette may have obstructed the light that should be passing through the cuvette. So with these foreign materials and markings on the cuvette, you may obtain a higher absorbance than usual. Next, foreign materials may have been introduced onto the samples when it was exposed to the atmosphere. A slight difference in composition can affect the absorption reading. Go. The last possible source of error is that the cuvette may not be cleaned properly in between trials. So it is possible that there may be small traces of the sample from the previous trial left in the cuvette, and when you add the new sample, its composition would change due to that small traces. Thus, you will have a different absorbance yield than you would normally have. We could avoid these possible sources of error by being careful and meticulous in conducting this experiment. So we must ensure that the cuvette must have been thoroughly cleaned and wiped before introducing new batch of samples and before putting it inside the spectrophotometer.
Considering the results that were discussed by Mr. Ramos, it can be concluded that the objectives for the experiments were done successfully. The absorbance of methylene blue was determined with 2.69% error, and also the results regarding the concentration and adsorption of methylene blue obeys the Beer-Lambert's law, in which they have direct proportionality. And also a linear equation was made, and it was used to determine the unknown concentration of a sample with a given adsorption. The method for the determination of methylene blue active substances with methylene blue on drinking water, surface water, and wastewater has been a standard from the ISO since 1996. As these methylene blue active substances are anionic surfactants that are being commonly used on cleaning products, it is harmful to the water forms, aquatic organisms, as well as the human body. And this way, the determination of methylene blue active substances in water samples is essential for water treatment.
The following are the references used for the experimental design. Thank you so much for listening, and good day.